Welcome to this webinar series from EUROTOX. As you know, EUROTOX has started to. Can you hear me well? Yes, I hope so. Everybody can hear me. It's fine. Okay, good. If you don't hear me, please put it in the chat. Okay, so it is working. This webinar series is once a month. So we had to cancel the last one that was concerning reproduction toxicity given by Sheila Price because she was sick. But we have the first one before, given by Corrado Galli on a journal introduction on toxicology and risk assessment. That was very well attended. And today we have the chance to have a seminar given by David Jones on how well the COVID vaccine developed so quickly. And I'm going to tell you some words about David. So David spent 8 years in contract toxicology, 11 years as a toxicologist in the pharmaceutical industry, and then he joined the MHRA, the Medicine and Healthcare Products Regulatory Agency in England, so in 1996. He very recently retired in 2021 from the agency after 25 years spending in the MHRA, telling you that it is very an expert in regulatory development of drugs and biologics. And so when he retired, he was a senior non-clinical assessor within the agency. So his role, mainly in the MHRA, his principal role was to assess non-clinical data for clinical trial authorization, application for marketing authorization, and for both non-biological and biological products, and also for advanced therapy medicinal products. And he assessed every UK application regarding COVID-19 since between 2020 and 2021. He also gave regulatory advice to the companies on behalf of MHRA. He represented the European Union in ICH revision of the M3 guideline and on the ICH S10 guideline, and also on the Q&A document for ICH S3, which is toxicokinetics, and ICH S11, which are the non-clinical studies to support pediatric medicine. So David is now working as an independent pharmacotoxicology consultant, and I must say that I went across several times with David in meetings, and when David is giving a meeting, it's always very interesting and passionate because he knows a lot about regulatory toxicology development. And I'm very happy that he accepted to give this seminar today. Thank you very much, David.
Thank you very much for that kind introduction, Mark. So I'll just share my screen now. So hopefully everybody can see that. Can everybody see that?
Not yet.
Okay. All right, because it's showing on my screen.
No, it's not on mine yet.
Okay, let's have a try again.
Yeah.
So if I put this into slideshow.
Oh, it's okay. And so if you have any question, forget to say, if you have any question, please put it in the chat and I will take the question after for the Q&A question. We can see it perfectly, David.
All right, just put it into slideshow. There we go. That should be better. So you can see that now.
Yeah.
Excellent. Okay, so thank you very much. So as Mark pointed out, yes, I did work for the UK's regulatory agency, and I did assess every clinical trial in the UK on to do with COVID, and I also reviewed all the vaccine data prior to their licensing. Well, certainly the first two. After that, I let other people do it. So any opinions I express in this presentation are my own. I do not represent the agency anymore in any way, shape or form. So let's take you back to 2019. When SARS-CoV-2 was identified as the cause of an outbreak of a respiratory illness in Wuhan, China. Now, I'm not going to go into how it got there. There are more conspiracy theories, etc., on how it actually got out into Wuhan. But let's just accept it did get out into Wuhan, China, December 2019. And it basically was causing a respiratory illness in people, and it was very readily spread from person to person. So I seem to have an automatic thing going here. On 30th of January 2020, WHO declared COVID-19 outbreak a public health emergency of international concern. And as I'm sure you're all very, very aware, things escalated from there onwards. Now, it appears actually that there's two distinct but overlapping pathological subsets of COVID-19, the disease. One's triggered by the virus itself, and the second one is a host response. The initial stage occurs at the time of your first exposure, first inoculation. And for most people, this was just a very mild and often non-specific symptoms, such as you felt a bit weird, you had a bit of a fever and a dry cough, and obviously at this stage treatment was just basically symptomatic relief. The second stage is when the viral multiplication is taking over, so an established pulmonary disease was brought in, and this was caused by a massive immune response as the leading symptom. This time, patients would be getting viral pneumonia, cough, fever, and possibly hypoxia. And very often, as you all know, a lot of people required a mechanical ventilation. Now, a minority of COVID-19 patients went into the third and the most severe stage of the illness, and this was basically a systemic hyperinflammation syndrome. The body started attacking itself essentially. So this is where you got cytokines increased, interleukins increased, people were going into shock, respiratory failure, cardiopulmonary collapse, and basically the whole body was just attacking itself. And this is obviously where the majority of people were dying. Now, it was well published at the time and is reasonably well known. A typical vaccine development takes about 5 to 10 years, sometimes longer, to assess whether a vaccine is safe and efficacious in clinical trial. Oh, dear me, I don't know why I'm going here, sorry. To go through the whole process and to manufacture sufficient quantity of vaccine doses for widespread distribution. So the first clinical trial authorization in the UK for a vaccine was approved on the 26th of March 2020, and the first authorization for a vaccine in the UK, or globally really, was the 2nd of December 2020. So you're looking at about 9 months essentially. And obviously a lot of people had a few raised eyebrows. How on earth did we manage to shave off 9 years development without compromising vaccine safety and scientific or ethical integrity? And I will tell you now how we did it. So normally, non-clinical testing of vaccine candidates typically start in animal models, first in small mammals such as mice or rats, sometimes non-human primates. And these are important for eliminating potential vaccines that are either too toxic or don't induce any indication of a protective immune response. And many vaccines that appear to be safe and induce protective immune response in animals actually fail in human studies, but only the vaccine candidates that are promising in these non-clinical testing move into the phase 1 clinical trials. Now, a phase 1 clinical trial, this is the first step in assessing a vaccine in people, typically 1 to several dozen healthy volunteers. And you're basically looking at soreness at the site of infection. Do you get a fever? Is there any muscle ache? But more importantly, is there an immune response here? Often they will be testing different vaccine doses, and only if a vaccine candidate is shown to be safe in these phase 1 trials will it move to a larger phase trial. But to get here can take anything between 1 and 10 years. Now, why is that? Excuse me a second. We'll come back to that in a minute. So phase 2 clinical trials, these continue to assess the safety and immune responses. You've now got a larger number and more diverse group of volunteers, typically up to several hundred people. These also include target populations of a specific age or sex, people with underlying medical conditions. Usually a vaccine for a child will start in an adult and then move down the age groups. And there'll be different types of immune responses will be measured here, including antibodies, cell-mediated immunity. But these phase 2 trials don't actually assess how well a vaccine works. They're just giving you a little bit more safety data and a little bit more indication that there is a good immunity induced. And to get here would usually take you about 2 to 3 years. And then we go on to the phase 3 clinical trials. These are critical to understand whether the vaccines are safe, more importantly, how they're effective. These often include tens of thousands of volunteers. You are chosen at random to get the vaccine or a placebo. And these are double blind, so the investigator doesn't know whether you're getting the placebo or the active, and neither do you. And then they're often followed up for quite a while to see essentially how many people get the disease and how many people are protected. And essentially these can take between 2 and 4 years. So that's where the sort of the whole 10 years thing comes from. Now, every country has a regulatory approval process for vaccines. In the UK, it was obviously the Medicines and Healthcare Products Regulatory Agency. And there was one of the things that the UK had, which was quite good, was Regulation 174 of the Human Medicines Regulations 2012, which basically enabled us to have a rapid temporary regulatory approval to address significant public health issues such as a pandemic. But scaling up vaccine manufacturing is typically done at the end of the regulatory process, because obviously this is a huge financial investment. So what are the major hurdles causing vaccine development to take so long? And it's quite simply, it is money and resources, because outside of a pandemic, you'll never make an awful lot of money developing a vaccine. And so, you know, I spoke long and hard to some of the people at the time, and they said, you know, you have to scrabble around to get a little bit of money to do your first non-clinical data. And then if you can actually manage to do the non-clinical work and you look like you've actually got something that's going to work, then you have to go out again looking for money so that you can just make enough vaccine to get into your phase 1 trial. And it's always this, you know, as soon as you get a little bit of data, you then have to go out and look for some more money. You have to look for someone who's actually got some resources to make the vaccine. You've got to find someone who's, you know, quite happy to go into the clinical trial. And the majority of that 1 to 10 years is actually taken up by that, is taken up by looking for funding, looking for resources, trying to get enough money to get to do a clinical trial, and then go away and do your resource to get your resources. But obviously you also have to understand the target right at the very beginning. You have to understand the virus, you have to know how it's acting, how it's getting into the cell. Manufacturing is a problem, you know, because there are quite a few vaccines out there, as you already know, and sort of try and get a slot to manufacture your new vaccine is incredibly difficult, or was incredibly difficult. And obviously just trying to get enough volunteers to do the take part in the clinical trials and to find testing sites. All these are very, very resource heavy. And so the actual running of the studies, the non-clinical studies themselves, they are very, very quick to run. The phase 1 clinical trial, you can get away, you know, after about, you can get through it within a month. The phase 2 clinical trials, you know, the actual running of the trials does not take long. And, you know, and to be honest, to run a phase 3 clinical trial with a vaccine, it depends on just how prevalent the disease is, because obviously you have to show that you are protecting people from catching the disease. So outside of sort of a pandemic situation, to actually get the proof that your vaccine is having a significant effect is quite challenging. So it's all down to money, resources, and things like that, rather than actually running the studies. Now, on the 26th of February 2020, we were approached by Oxford University, had a very nice meeting with them. They were talking about this problem that was in China, and that, you know, this new disease that was in China. And Oxford University had already been working very long and hard on two very, very similar viruses. One was for MERS, which was Middle Eastern Respiratory Syndrome, and one was SARS, that was Southeast Asia Respiratory Syndrome. And so they said, you know, we've got a lot of data on MERS and SARS. Essentially, this is yet another coronavirus. What is happening to my computer here? Sorry. Can we actually use the data we've already got to actually sort of to support going into this COVID disease? And, you know, I looked at it, and we'd already had similar discussions with them over various other platform technologies that they were having for various other diseases. And we agreed that the use of platform technology and any previous data they had was a very, very acceptable approach. And we said, you know, Barry, go away and do it. Because essentially, what people tend to forget is that COVID was actually the 9th coronavirus that passed from animals to humans. So there was a lot of understanding about it. And you can always look at a sort of a vaccine, almost like a sort of a Lego block, where you just tick off one little colored piece and put on another colored piece. You know, the majority of the vaccine is standard, or certainly was in those days. When we were talking to Oxford. So we had this sort of a plug and play vaccine manufacturing. So it was very, very quickly that you could actually get a new vaccine designed. Now, so it started off with designing this vaccine, you know, because obviously designing the vaccine begins far earlier than the jabs in animal stage. And so the first stage was deciphering the exact genetic makeup of the SARS-CoV-2, the virus that was causing COVID-19. But by early January 2020, that genetic blueprint was in hand. It had been published. Chinese had sorted it out. And so we knew from January 2020 what the virus looked like. And just to put that into perspective, you know, I mean, everybody knows that the entire human genome has been mapped out now. That took about 13 years, starting in 1990. But thanks to the computers that we have now, the same task can be done in a matter of hours. So from the first patients that were actually having COVID in Wuhan back in November, December 2019, to actually knowing the genetic blueprint of the vaccine took about 2 months. But most crucially, the researchers had this genetic instructions for making a spike protein. The protein, how does that virus use to get into the cells? And so this is a key ingredient for making the vaccines. A lot of things in the press at the time about the spike protein. And these spikes basically jut out from the virus surface. They are proteins, and they make a very, very easy target for the immune system to recognize. And so researchers already knew how to zero in on these proteins, thanks to decades of work studying other coronaviruses, including SARS and MERS, like I said. And so much work had already been done, it was relatively simple then to, I say simple, it wasn't that simple, obviously, but in the global scheme of things, it was relatively simple to design a vaccine to stop the virus fusing to the cell. So in February 2020, after chatting to us, the Jenner Institute of Oxford University actually got a grant of money to produce 1,000 doses of the vaccine for the clinical trials. Now, their original intention was basically to donate the right to manufacture and market the vaccine to anybody who wants to do so. But the Gates Foundation, Bill and Melinda Gates over in the States, they actually urged Oxford to find a large company partner to get COVID-19 to the market as quickly as possible. And the UK government basically encouraged Oxford to work with AstraZeneca. Agreement was set up, and that's how it became the sort of AstraZeneca-Oxford franchise. But in April 2020, obviously, we now had a global pandemic. It was no longer a problem just in China. And so the UK government set up the Vaccine Task Force. You might have heard about that. It was in collaboration with Patrick Vallance, the Chief Scientific Advisor, and Chris Whitty, the Chief Medical Officer. And basically, the thing here was it was designed to facilitate the path towards the introduction of a vaccine into the UK and the global distribution. And this task force had a lot of people from industry backgrounds and from various other business backgrounds. And they managed to, well, they were tasked to coordinate the whole research efforts of the government with industry and academic and various funding agencies, just to sort of speed it through. So Oxford and other people were now in a nice position where they didn't have to go out and look for money. The money was basically dangled in front of them, said, if you want money, here it is. The manufacturing processes were all geared up for them. So the task force overall strategy was not to concentrate on one vaccine, but to have a diverse portfolio of vaccines with an emphasis on those capable of achieving an immune response in the over 65s. Now, we hear so much about the Oxford vaccine, the AstraZeneca of Oxford. We hear a lot about the Pfizer-BioNTech mRNA and a few others. There are actually about 200, there were just under 250 potential vaccines flying around to begin with. So the task force homed in on 6 candidates, which employed 4 different methods. There was the adenoviral vectors, the mRNA vaccines, which were coming through then, adjuvated proteins, and there were a few whole inactivated viral vaccines. So they homed in on 6 and funded those 6 to go forward. So initially there was a not-for-profit licensing agreement signed between Oxford University and AstraZeneca, basically to make up 1 billion doses of a potential supply secured, and the UK reserved access to an initial 100 million doses. America reserved 300 million doses, as well as giving authority to perform phase 3 trials in the States. So again, the manufacturer, sort of the developers, didn't have to sort of look for who was going to buy their products. They already had 400 million doses reserved, if you like. So the manufacturing places were in sight, and money was no longer an issue. And this was the major factor on why it was able to move so fast. And there were some other factors, and we'll come on to them in a minute. But essentially, the collaboration got £68 million from the UK government. They got about $1.2 billion from American government. So clearly, money was no longer a problem. And on 4th of June 2020, AstraZeneca announced that they were going to give it $750 million coming in from COVAX program. That was to develop the vaccine throughout the world, and 300 million doses were going to be distributed to low-income or undeveloped countries. But then sort of moving backwards, obviously regulators weren't just sitting around waiting for people to do something. And on the 18th of March 2020, there was a teleconference of global regulators convened under the auspices of the International Coalition of Medicines Regulatory Authorities, ICMRA, and the WHO. And we were discussing the regulatory considerations on how to get these vaccines into the first in human trials as quickly as possible. So I represented the MHRA from the non-clinical viewpoint in this, and there were people there from virtually every regulatory authority in the world. And I was asked to outline just the work we'd already done, and to discuss platform technology. And so I outlined how, we were using platform technology to support those first in human trials in humans. Basically, everybody acknowledged that, the extent of non-clinical data to support going into a first-in-human clinical trial would really depend on the vaccine construct, just how much data was available for the construct, and data from closely related products. And although mRNA was a relatively new way forward in those days, people already had mRNA vaccines kicking around. They were being developed for other respiratory problems. There were mRNA vaccines in development for influenza. So again, they were able to build on the data they already had just to tweak it and move it forward into COVID. And everybody at this meeting recognized that obviously there was very limited availability of non-human primates, because a lot of authorities at that time were insisting that vaccines had to be tested in non-human primates. But everybody agreed that, maybe we could actually start to look away from the non-human primate and look for better animal models anyway, to avoid delays going into clinical development. Now, it wasn't unanimous, but basically, most people agreed that some of the vaccine constructs where there was a lot of information kicking around, that you could actually get into the first in human trials without conducting the safety studies in animals, as long as you had proof that the things were actually producing an immune response in an animal model. You could actually go into the first in trials in humans without that animal safety study, providing that there was lots of risk mitigation strategies in place. And then that first in human trial would be running parallel with the safety studies in animals. So again, this was a very significant saving in timing, because you wouldn't have to do sort of a 3 months of animal testing before that first in human trial. They were allowed to do the first in human trial while the animal studies were ongoing. Now, the WHO also set up quite a few ad hoc expert working groups, one of which was focused on disease modeling. And the goal of this was for people to work together to identify a good animal model that would actually we could work. And the good thing is this was helping to reduce duplication of efforts. Everybody was working together rather than everybody working individually. So again, this was really great, and obviously fell into the 3 R's as well. Having said that, a suitable animal model remained elusive. Basically, you want a model that is you can infect with COVID very, very easily, and then reproduces the clinical course and the pathology observed in humans. And this actually proved to be very challenging, but we moved forward anyway. One of the best models that was coming out was the K18H-ACE2 transgenic mouse. This was quite a good little mouse model. It actually had all the severe lung damages and the neuronal damages. Didn't actually produce sort of some of the side effects as well. Did show good replication of the virus in the lungs, did have weight loss, did develop pathological lung inflammation, and died within 4 days of post-infection. So this proved to be a very good model to test the vaccines out on. This golden Syrian hamster was also shown to be a reasonably good model. You could actually show that transmission from naive to co-housed animals actually spread the disease. The animals generated neutralizing antibodies. The significant drawback with these was that they got basically they recovered very, very quickly on their own without any vaccine. But it was a reasonably good animal model to use. Non-human primates, they were actually used as a model to study most of the vaccines. They did develop a disease very similar to humans, but essentially they never developed the acute lung injuries. So they only were sort of mimicking the sort of the mild disease that people were just recovering from with quite easy. And I was very much against people using the monkey. I didn't think it was a good model. Some other agencies disagreed with me. So although in 2020, we were still part of the EU medicines regulatory network, thing about clinical trial authorizations in Europe, there are national decisions. So basically we were quite able to induce significant flexibilities ourselves. We didn't have to refer back to the EMA to say this is what we want to do. And we worked very closely with some other UK government agencies and healthcare partners. They didn't displace or diminish any other obligations applicable to relevant products, but it just meant that we were able to identify various things and to sort of just to make some timing savings. We also provided scientific advice and rapid review times. We actually turned around clinical trial applications within 48 hours. It usually takes about 30 days. So, I was working 15-hour days, 7 days a week. But, the whole thing is that we were just everybody was working flat out to get these clinical trials authorized and running. Now, the National Institute of Health Research also played a critical fight in the UK. They were funding and delivering groundbreaking research that basically was helping to save lives. The good thing about the NHS, what they were doing is they were identifying, if you like, hotspots where the disease in the UK was very, very prevalent, and basically telling people, okay, this is the best place to test your vaccine. There's a lot of people out there. So you've got a better chance of actually finding a positive result. And they were, in finding sites for people. So again, this meant that the people who were developing the vaccines didn't have to go around looking for sites. Basically, they were given a list of sites, so they were able to respond very, very quickly and get the studies up and going. Now, nicely, the pharmaceutical companies were working globally, and there was an unprecedented level of cooperation, not just in sharing science, but actually in sharing manufacturing processes and manufacturing sites. And this meant by actually March the 25th on 2021, it was basically less than a month after the pandemic was really going globally, the USA's ClinicalTrials.gov website listed 5,185 clinical trials ongoing for COVID-19, including 528 vaccine trials. So, unprecedented amount of work was being carried out trying to tackle this disease. Now, the UK Vaccine Task Force, they also identified manufacturing sites and they secured access to sites. This gave the UK permanent capabilities because we actually built a new site as well. A lot of controversy about this because I'm sure you people in Europe will possibly remember that some of the vaccines were manufactured in Europe and were being merrily shipped to the UK rather than being used to vaccinate people in Europe. But it was just the UK Vaccine Task Force has done their homework, if you like, and tied in these places with very good contracts. But at the same time, the UK actually built 2 sites and expanded 2 sites. So basically, the capability to manufacture the vaccine, whatever format it was there. So there was a manufacturing capability was set up before the vaccine was actually finalized. You may remember one of my slides earlier on that said this basically you wait till you've got a vaccine and then sort out to manufacture it. But the manufacturing capability was actually in place before the vaccine was finalized. Now, the MHRA for many years provided scientific and regulatory advice to sponsors. It was a major part of my job. We allowed people, even before COVID-19, you could come to the MHRA at any stage of your initial development to have a meeting But we offered a free regulatory advice meeting for anybody developing a COVID-19 associated product. And I do mean anyone. You didn't have to be a company or a hospital. So we did have some very strange meetings. And essentially in 2020, I actually had over 240 meetings with companies and academic institutes and hospital groups, all to discuss COVID-19. And our clinical advice helpline was fielding about 1,000 queries a month at its peak in 2020. So by meeting with companies, telling them what they had to do, looking over their data before they actually submitted it to us, it formally, all managed to sort of cut down the development times. And so we were the first regulatory agency to formally approve a vaccine against COVID-19. And it wasn't just the MHRA, we still went through all our advisory bodies, but essentially we also introduced the rolling review. So we'd actually, although the vaccines were finalized and authorized in December, we'd already started reviewing all the data from October 2020. And we did the same thing for clinical trials. It sounds very impressive turning around a clinical trial authorization in 48 hours, but we'd already seen the protocol. We'd already seen early versions of the investigators' brochures. So we had this rolling review going through, which just meant, the final stages were done in 48 hours. But like I said, we'd already seen greater majority of the data before we got there. And again, all this just cut down the whole process, because if they'd, in the sort of normal circumstances, they would have put in their applications at the beginning of December, and we would turned it around in 150 days. But because we'd already been looking at most of the data from October onwards, it meant we were able to turn around the final authorization so quickly. But like I said earlier on, another resource is usually trying to get volunteers to go into these clinical trials. There's usually a shortage of people who want to be a volunteer in any sort of clinical trial. But in the UK, the NHIR and the NHS set up sites for people to sign up in advance of trials, and almost 300,000 people volunteered to go into the clinical trials, which again, this made recruitment for the trials far quicker and obviously significantly reduced the startup time. So again, there was not this delay in, all right, I've got my vaccine finally, I've got my clinical trial authorized. Now all I've got to do is find some people. The people were already there, waiting to join, waiting to go into the clinical trials. So this is basically why we were able to get through in 9 months instead of 10 years. It was all down to the fact that money wasn't a problem, manufacturing wasn't a problem, investigator sites wasn't a problem, volunteers wasn't a problem, and the regulatory authorities were far more flexible. So it just meant everything was quicker. Now, another reason, usually in phase 1 clinical trials, you will test several doses, but essentially they did to a degree, but it was a very limited number of doses that they checked out. And even in the phase 2 clinical trials, instead of trying to find, the fantastically optimum dose and the optimum testing regime, or, because it's always a sort of a prime boost sort of situation, it was basically, okay, well, this is working, this is giving a good immune response. Yeah, it could be better, could be worse. So that's where, again, instead of having to sort of do an awful lot of testing to make sure you had the perfect dose to go into the phase 3 clinical trials, it was basically, all right, this dose seems to do what it's supposed to do. Let's get into the clinical trials. And then even in the phase 3 clinical trials, there was a interim analysis set up. So as soon as it looked like, all right, we think we've got enough people now showing that they've had the disease, let's have a quick look to see how good these vaccines are. The WHO had already said, okay, we just need a vaccine that will give you, will protect, say, 50% of the patients. That was the sort of the minimum criteria. As it happens, the vaccines had a far better efficiency than those. And so, yeah, it was just a question of throwing money at these things, and the fact that people weren't having to sort of scrabble around looking for resources. That's why it was, we managed to get these vaccines tested and approved within 9 months instead of 10 years. So that's about it. So I'm finished giving you my talk. And now I'm more than happy to take any questions you might have. Thank you very much.
Thank you very much, David, for this excellent presentation. Very clear. Now we have all the explanation. So I have a first question from Renata Lochner. I'm sorry for the pronunciation. From EFSA. I mean, she's wondering about the role of non-animal testing methods and NAMs in this context. Are these lessons learned and potentially increasing potential for these new approaches identified also in the future vaccine development? So the idea is the place of NAMs in future vaccine development.
I mean, yes, we did see an awful lot of non-animal work applied. But a problem with a lot of the non-animal work is that they are, if you like, not, well, they're not 100% validated. I mean, nothing's 100% validated. Even the animal models, aren't perfect. But it was very much a case of, well, let's stick with what we know. We've, developed vaccines this way for quite a while now. It is a sort of a very established way of going. But yes, I had a lot of meetings with people coming in with suggestions for non-animal models. We encourage people to do those non-animal models because animals were at a premium anyway. So we did see an awful lot of non-animal methods. And I think, again, it'll be lessons learned. And I think people will be building on these methods to go forward. And yeah, so again, the non-animal methods, I think we will see more and more of them as time progresses.
Thank you. There is also another question from Dr. Anetor. On the question is, how did handle vaccine hesitancy phobia? I mean, it's.
Sorry, how did?
Handle. So can you precise your question, Dr. Anetor? It is on during the process of development, they had to.
How did we handle vaccine hesitancy or phobia?
Exactly.
Yes. So, I mean, this is a good question. And, obviously, one of the massive problems we had was the anti-vax people were very, very vocal. An awful lot of false data put out there. It was quite funny. There was a post came out from Russia, which basically pointed out that the Oxford vaccine was basically based on a primate background. And this post basically said that anybody who would take the Oxford University AstraZeneca vaccine was likely to change into a monkey. And there was so much, I mean, you probably saw the protests and things. There was so much anti-vax things coming out there. And again, a significant amount of my time was dealt with sort of writing, responding to various people who had these problems. And a lot of it was down to the fact that, the press had made such a big thing. Oh, it's going to take 5 to 10 years to get a vaccine. And, hang on a second, we've got a vaccine within a month. So there was a lot of that trying to sort of put it over to people why we got where we are. We weren't cutting corners on all this. And I think it did help the fact that, from the phobia hesitancy point of view, so many people were dying worldwide. You have to remember that thousands and thousands of people were dying worldwide. So that is quite a sort of incentive for people to have a vaccine. So I think if it had been a much rarer disease, and if there had been, it wasn't that fatal, then I think we would have obviously had far more hesitancy. But as it were, I mean, the majority of people were very, very keen to take the vaccine. Like I said, 300,000 people signed up as volunteers before we'd even got the first vaccine into clinical trials. So we were able to get around that one. So the next question was the inclusion of women, pregnant women and children. Did they use animal studies for those questions? Certainly for the pregnant women, yes. There was reproductive toxicology studies were done on that. But there was very much a benefit risk thing coming in, because it was already showing that women who had COVID-19 were miscarriaging. So again, there was we had a very, very clean vaccine. Scientifically, there was no reason to believe that the vaccine would cause any problems for a pregnant woman, and it came down to risk benefit. But we did ask for very, very short-term teratology studies to be conducted. These studies were conducted, so that's before it was actually approved, if you like, for pregnant women. For children, again, the reason we were able to go into children is because we had a lot of adult data, animal data for children. You don't do studies in juvenile animals for a vaccine. It is all it's okay in a human, and then we just used the human data to sort of develop the proper dosing regimens for children. Now, you might remember at the time there was so much discussion on what was the correct timing for the prime and the boost, and there was initially it was set at a certain time and then it changed. Some of that actually came out because there was a mistake in one of the clinical trials. It actually missed the timing when they were supposed to give the they gave the boost laced and found out, oh, actually it doesn't. It actually has a better response if you are delayed slightly. So that was quite fortunate in ways. So, but again, the majority of these clinical trials were basically built on previous designs, and it worked. Therefore, we didn't need to go and for the optimum. We just needed to get the vaccines out there as quickly as possible.
We have a question on the animal model, because you just described the different animal models, and there were a discussion at one point on the utility of the monkey studies, because the monkey, they get infected only on the upper tract and they don't get very sick. But there were some agencies that were very, very, very pushing much for the monkeys, because I don't know, it's more close to human. So your proposal was.
I mean, yeah, absolutely. I had quite a blazing row with our friends across the Atlantic, because they were pushing for the monkeys all the while. I was saying the monkeys were only replicating the very mild disease, and not trying to be callous here. We didn't care about people who were getting the mild disease. The people we were really worried about were the people who were dying for the severe disease, and therefore we needed a model that actually matched that. But basically their argument was, well, if it helps with the mild disease, it will probably help with the severe disease anyway. So they kept pushing for the monkeys. That's why there was a global shortage of monkeys. It was really that there were certain agencies that were adamant that we'd always use monkeys, therefore we're going to use monkeys. So America, China, these agencies insisted on primate studies. Other agencies, certainly the UK, and our friends across the English Channel, were far less rigorously asking for the monkey. We just wanted a good model. So fortunately for monkeys, not for golden serum hamsters. The golden serum hamsters seem to be a slightly better model. Again, like I said, although it did replicate the disease a lot better than the monkey did, they were able, they got better on their own anyway. So it was very, very hard to prove that the vaccine was doing a lot. Because if it was going to cure itself in 14 days, our vaccine, it was very more problematical. But as I always tell people, the fact is you don't use animal models to prove that your vaccine is safe. You do not use animal models to prove that your animal models to prove that your vaccine is going to work in humans. You do these animal models to show that it's acceptably safe to go into humans. Because so many, not just vaccines, so many drugs, we can probably cure every disease known to man in an animal model, but you go into humans and you have a very different thing. So I wasn't desperately insisting that there was fantastic animal data. I just wanted those vaccines to be sure that yes, they were producing an immune response. Yes, there was no huge safety concerns. Let's get them into humans and let's see what happens there.
And I've got one more question, because one more question on about the. Okay, so it has been developed very, very quickly. So it's a question of science, because you already told us that science was already there. There were a lot of lessons, and sequencing was very fast. So the amount of money was incredible, which can help, of course. But also there have been, and you just told us about the cooperation between the different agencies in different countries were really optimal in some way, which is not always the case during drug development. So do you think that the principal criteria for this fast approval is a combination of the three, or one of the three was better, or it's really a combination?
I mean, if I'm going to be cynical, I say the most critical was the fact that money wasn't an object. And I've spoken to lots of people ever since, and that researchers said, even working for companies, even companies that were specializing in vaccines, in normal times, it was very, very difficult to persuade senior management to give you the money to do the studies you wanted to do. So I think the fact that money was no longer an object was a massive thing, because that's what a major disease. I spoke to Andy Pollard, I think it was last year. Andy Pollard was one of the investigators who was from Oxford University, and he was saying, that the amount of time that when he was doing the SARS and the MERS vaccines, it was just a question of having to persuade people that this was quite serious, and I do need a little bit of money, and they were just given the bare minimum amount of money to do the work, and then once they've got the work, then they would have to go back and look for more money, more resource, and that, he says, has always been the major hold up, the fact that you couldn't get a hold of money to do the work, and the fact that manufacturing was trying to get a manufacturer to sort of manufacture 1,000 vials, just for your one study, was very, very problematical, because they wanted huge protocols. So money and manufacturing, the fact everybody scaled up their manufacturing, I think those were probably the two major things. But yes, there was an unprecedented amount of cooperation between companies. A lot of companies worked together, pooled their resources, and the amount of publications. Everybody was not hanging on to the data; they were flinging it out into the public, into the scientific community as soon as it was developed. I mean, the Chinese could have held back on that genetic makeup of that virus, and just held it for themselves, but it was out there in January of 2020. So just an unprecedented amount of cooperation. And we've learned an awful lot from those. And you'll find that a lot of the regulatory authorities now have brought in the rolling review. The MHRA offers it as almost as a standard now. Yeah, you have to pay extra. But, there was just ways of cutting down the regulatory delays, which everybody is very, very happy with. The cooperation between regulators and industry has improved enormously. There was always in many agencies, there was sort of a reticence to sort of give scientific advice, to talk to companies, because you were seen as basically in the pocket of the pharmaceutical industry. A lot of criticism had been aimed at the MHRA over the years, because we do give scientific advice. I say we, I'm not there anymore. So if you're telling people how to do something, then, when they do that, if they, you automatically accept that that's the right way of doing it. So there's so much negativity about helping the industry that regulators should be very much arm's length and shouldn't be out seen to be helping people. A lot of that has gone now. So a lot of people realize that the more regulators and industry talk together, the smoother things are and the faster products get into patients, and that's what we want. So but politics came into it without a doubt. I mean, I'm sure you all remember the French president and the German president basically saying, "Oh, you can't give this vaccine to the over 65 because it hasn't been tested." And that didn't just have an impact in Europe, because a lot of the African nations who still have quite close links to some European countries, when they saw that Europe was turning its back on the AstraZeneca vaccine, there was a sort of an undercurrent to say, well, hang on a second, if this vaccine isn't good enough for Europe, why is it good enough for us? So there was a pushback from a lot of the African nations. Even though the vaccine was there, they couldn't persuade people to take it because they'd read in the press that the Europeans didn't want this vaccine, didn't want, to be fobbed off with a vaccine that wasn't as good as other vaccines. So, but those nations couldn't afford the mRNA vaccine. So politics was quite a problem as well.
So this is what your answer was relating to the last question that you can see on the chat.
Oh, yeah.
Yeah.
Yes. Yes. So it is very, very difficult. I can criticize the UK government in some ways because the UK government at the time very much said this is a British success. Britain is fantastic. We've got the vaccine out so quickly, and they just basically were flying the flag everywhere. And let's face it, Brexit was still a sort of an open wound, and a lot of European countries were sort of almost had a sort of a if it's good enough for the British, we don't want to know. So politics, unfortunately, is a huge problem. I think the UK and Europe are working a lot closer together these days. So hopefully that won't go away. There was no doubt, I think, in a lot of people's mind that the FDA were under a lot of pressure only to authorize American vaccines. Absolutely no idea. Janssen's had massive problems trying to get into America. AstraZeneca did, but Pfizer, Merck, they had no problems whatsoever. So, and Moderna, obviously, these were American products. And yeah, there was, the cynical side of me, looks at it and think, why were the FDA, going hell for leather for the American products at the expense of European products? So, yeah. We'll forget about the Chinese and German and Russian vaccines because we won't go there. But yeah, so yes, unfortunately, politics was very big. I mean, you had Trump at the time. It was Make America Great. So I think he wanted an American vaccine without a doubt. So yes, but I think fortunately the world is sort of is more cynical towards politicians these days, and hopefully that won't be a problem in the future.
I hope so. So there is no more question yet, and it's 1 o'clock. So I really again want to thank you very much, David, for this very good presentation and this very nice exchange with the people and a very good question. So I've got a comments also that is many thanks for a very interesting talk. With the established vaccine now and the milder variants, this looked like already ages ago.
Yes, yes, it does, doesn't it? So, and it has changed. mRNA vaccines are coming forward in increasing numbers now. That's upsetting the anti-vax people more because they are seen as you're introducing foreign genetic, but yes, we can hopefully get around the anti-vaxxers.
Hope so. They're very strong in France.
Okay.
Thank you
Okay,
again, David.
so thank you very much again for inviting me,
Thank you.
and
And
have a
thank
good
you.
day, everybody. All right,
Bye-bye.
bye-bye, everybody.